So first of all, where we're from. It's very important in New Zealand to show where you're from. We're from the University of Otago, which is in the South Island of New Zealand for those visitors here. And we have 3 campuses out. This is showing the main campus. We have beautiful spring in Christchurch, and in the South Island generally, so we have wonderful cherry blossoms. If you come in September. It's a beautiful time of year. But we're in Christchurch, which is in the centre of the South Island, and we are a medical school. And at that medical school, we have a huge research community, and as part of that, we form the Centre for Free Radical Research. So we're not a political organisation. We are a research group that specialises in the measurement and understanding of the mechanisms of disease. So because we're at we're on a health campus, we're interested in human disease, what causes it, how to prevent it, and therefore we're also interested in human nutrition. So to start this talk, I'm just going to recap a little bit on Professor Siedel's talk from yesterday. Now, we all know that we're very lucky these days because we have the good news for us is that we have much greater life expectancy than many of our predecessors ever had. And so this curve has been going up since about the 1850s. And that's partly due to better nutrition. It's also due to better nutrition for children, so our infant survival is a lot better, and that contributes a lot to overall survival. And also to our ability to fight off infectious diseases. So vaccinations and antibiotics have contributed enormously to our increased lifespan. But we know that increased lifespan comes at a cost, or we think it comes at a cost. So we have the preconception that as we age, we're going to lose function, and that we have increased risk of exposure to non-communicable diseases, and these are certainly on the rise in our populations. So we now fear, as we get older, the onset of cardiovascular disease, cancer, diabetes, cognitive decline, and frailty. And so our emphasis with respect to health research is not on increasing necessarily our lifespan, but increasing what we call our health span. So how much of that life, for how much of that life are we going to be able to be healthy? It's not necessarily a given that we're all going to get all of these things as we age. In fact, if we look at some of these non-communicable diseases and the causes and risk factors for them, we all know some of us have a propensity in our genes to end up with cardiovascular disease or for predispositions for different types of cancer. But however, even though there's a lot of emphasis put on that, the contribution of genetics is actually quite small. And there are much bigger contributions, shown over on your right, of diet and lifestyle. And so the size of those balloons suggests how much of these diseases is modifiable by diet and lifestyle. And so you see here, from this data from 2002 in the US, that 70 to 80% of colon cancer, stroke, and coronary heart disease is preventable by diet and lifestyle. Type 2 diabetes, 90% preventable by diet and lifestyle changes. Now, we know this is possible. One of the reasons that we're here at this conference is how do we achieve that? And that's one of the things that we like to consider here. Now, what Professor Siedel referred to yesterday was the overwhelming evidence that we have that a diet rich in fruit and vegetables correlates with decreased risk of many non-communicable diseases and also with overall mortality. Now, there are huge epidemiological data for this. And much of it is shown in studies like this, where we analyse an association between portions of fruit and vegetables and risk of death from any cause. So all cause death, if you look at the numbers, just look at these numbers across the top, then you see that as you increase your number of fruit and vegetable serves per day, your risk of all-cause mortality and cardiovascular disease goes down to almost half of the level that you might have had just by eating a much better diet. Now, we know this from studies like this around, so these are massive kind of epidemiological data that we have. We also have the similar kinds of analyses for some specific diseases. And so here's a correlation of fruit and vegetable intakes with metabolic syndrome, which is a predisposition to diabetes. And so you see the serves of fruit and vegetable intakes, or the quintiles, and so you see the more fruit, this is actually just fruit on this graph, so the more fruit you eat, the lower your biomarkers are for obesity. Your healthy fatty acids don't change at all, so your HDL levels don't change. So your serum triglycerides, glucose levels, and blood pressure all trend to a much healthier profile as your fruit and vegetable serves increase. Now, we have good epidemiological data about fruit and vegetables. And one of the reasons for that is that the kinds of studies that have been done with fruit and vegetables tend to measure exactly those things, high intake versus low intake. And so we see very good correlations. And so in the analysis of all of this mega data, we need to have our wits about us, essentially. And we need to think, are the studies that we're analysing, have they been well done? Are we analysing the right things? And are we able to actually measure an association with what we're doing? And so we consider, with all of these studies, study design. So how many participants have we looked at? Are we doing intervention studies or correlation studies? What's the frequency of measures? So, so how reliable is the measure that we're making? And all of these things are important in terms of analysing the impact of these correlation studies. So what do we conclude from that? Oh, actually, no. And so we will analyse mega data. So there's a trend now there are so many studies that we try and bring them all together by doing systemic analyses. And so all of those considerations about study design all need to be taken into account, and so we can group all of that data together. Now, by analysing all the fruit and vegetable studies and all the dietary habit studies, we can now come to the conclusion, and this is a pretty good consensus around the world, and Professor Seidel also referred to this, that higher fruit and vegetable intake is associated with decreased risk of chronic diseases, with decreased all-cause mortality, increased lifespan, and improved health status. So all of these measures, by and large, hold up in most of the studies on fruit and vegetable intake. So what do we recommend as a result of that? Well, we recommend that people eat more of these things. And so this is the US recommendation currently, half your plate, so half of what you eat should be fruit and vegetables. So this is trying to get the message across to people in terms of how much of these things you should be eating. We're no longer talking serving size, we're talking a proportion of what you eat. And that recommendation does not equal the reality, as we all know. Now, why? Why do we have this recommendation? What is it about, and the question was asked yesterday, what is it about fruit and vegetables that actually gives them that health-promoting property? And this is the thing that we're interested in. And what fruit and vegetables contribute is not macronutrients to our diet. We don't get protein, we get some sugars, but we don't get the macronutrient building blocks in our bodies. But what we get is we get micronutrients. When we're talking about our micronutrient need, we're talking about 13 vitamins and 14 minerals. So vitamins are things that our body can't make and that we need to get from our diet. And so they're listed over on the right. Some are water-soluble, some are fat-soluble, and they're not all the same. So vitamin intake, they all have different functions. So these micronutrients support many functions in the body. They support hundreds of enzymes that do all kinds of biological functions that we need for good health. They support heart health, immune health, bone health, energy production, normal cell metabolism. So we need it's like this is what keeps our engine running. So we need these micronutrients from fruit. This is what we get from fruit and vegetables. So if we're looking at those components, this is where we run into trouble with trying to make recommendations to people based on epidemiological and intervention studies. So this is an example of a meta-analysis or a systemic review of all of the studies that have been done into antioxidant vitamins. So we're talking about vitamin C, vitamin A, and vitamin E. 2 are fat-soluble, 1 is water-soluble. They're not the same thing. We're also talking about selenium and beta-carotene. Now, in this meta-analysis, and I put it up because don't try and read it, but essentially these people started with 16,000 references from the literature. They've excluded all kinds of studies, and they've ended up with 68 trials that they've analysed from 385 references. So 385 references of 16,000 that they started with. You look at the effects that they see over here. You see, if you're looking left and right of the line, any dot on the line shows no effect. You see some studies show an effect, some don't show an effect. What can you possibly conclude from a meta-analysis like this? One of the problems is that you can't conclude anything. And the reason you can't conclude anything is that even though you've taken this massive lot of data, you actually have no consistency between the studies that you're comparing. So if we look at that paper, which is a major publication, that came out with the conclusion antioxidant vitamins do nothing for health. We see that in all of the studies, they administered an antioxidant supplement to populations, but the doses that were used, some studies used the antioxidants alone, some used them all together. The doses ranged anywhere from, for example, vitamin A, 1,300 to 200,000 units. The intervention times ranged from 28 days to 12 years. The follow-up times were 28 days to 14 years. And these are the studies that have all been included, and you hope to come out with some single conclusion from that. Well, the reason that that's not possible is because, all these vitamins do different things, and so comparing one with another isn't necessarily going to give you the right outcome. Secondly, largely, the participants are healthy, and so there's no room for improvement in any of the outcomes that they're looking at. And also, they're giving supplements, and supplements tend to boost people over and above what we know to be healthy levels, and that, as we'll speak about later, really has nothing to do with the function of any of these antioxidant vitamins. So it may take you anywhere in the range of optimal activity to over and above optimal, and that really doesn't give us any information. And so there's a mistake on this slide, sorry. So our conclusion to date from supplement studies is that supplementation of the healthy population has no beneficial effect, on overall health status, on all-cause mortality or susceptibility. And this is the message that then goes out to our general consumers, who then get very confused and say, are we supposed to be taking these things, or are they good for us or not? And part of the message is based on what we think now is very flawed science with respect to individual nutrient supplement data. So some supplements, in fact, vitamin E and vitamin C and vitamin A, may in fact be harmful. And these are the fat-soluble vitamins that your body stores. And so we know that there are toxicity levels associated with that. So we're here to talk about vitamin C today. And so for the rest of the talk, I'm going to concentrate mostly on that. With the supplement studies, that's one thing. Those studies in which people have measured, like the fruit and vegetable intake, they have measured quintiles of plasma levels of vitamin C. So if you're looking at all our body levels, then those people who have low vitamin C, comparing people who have low vitamin C levels with high vitamin C levels, those studies almost unanimously show decreased risk of chronic diseases across the quintiles of vitamin C status. So measuring intake is not enough. You need to measure body status, and that will give you the information as to whether or not this is a benefit. And so there's just some examples here where serum vitamin C or plasma vitamin C levels correlate with a number of health status like blood pressure, overall mortality, and also susceptibility to some cancers. So those studies in which we have measured levels actually show an association. That's a different thing than the supplement studies. So now we're going to come to what do we know about vitamin C, and why are these cohort studies important, and why are plasma levels of interest? So this is what most people know, and for most people, their knowledge starts and stops at this point. And so we know that it's an essential nutrient, so we need it, it's a vitamin, we need it from our diet. It was discovered 100 years ago in response to a long search for the cause of scurvy, for the molecule in fruit. So James Lind in 1753 discovered that if you gave sailors fruit while they were sculling on the boats, then they didn't die of scurvy. But it took hundreds of years before we discovered what that molecule was. And it was vitamin C, which is a small water-soluble molecule. It's a small acid, and it's made from glucose. So this was almost a miraculous discovery. How could something so small and simple, very much like sugar, how could that actually prevent this devastating disease? And so most of the focus has gone on determining what it is about the deficiency syndrome that gives us the disease, and how does that relate to our health benefits. So what do we know about vitamin C and taking it up into our bodies? Well, we know we get it from food. And I want to concentrate a little bit on these plasma levels. So we know, depending on how much we take up, our plasma levels will range from 10 to 100 micromolar. From dietary intake, you won't increase your plasma level beyond 100 micromolar. We know from there that our tissues concentrate, all the cells in our body concentrate vitamin C from the plasma. So the plasma is a delivery mechanism, and it delivers vitamin C to our cells that actively transport it and concentrate it from the plasma to reach millimolar concentrations. And so across the different organs, we have in the brain up to 10 millimolar, so over 200 times what's in plasma in the brain. Now, this is an indication that there's some essential function in the brain cells. Similarly, in the adrenals, in the liver, in muscle, all the tissues of our body, except for red blood cells, concentrate vitamin C. So these were the signals. Why is it there? Why is it more in some places than others? And so we think that reflects a functional requirement in those places. These are the clues that we have. The other thing with the plasma level is how dependent are we on that delivery mechanism? I'm going to go over this very quickly. So essentially, it's pretty hard to get hold of that brain tissue from, most people are not willing to donate bits of brain tissue or even bits of liver. And so we've modelled the plasma concentration dependency on the tissue distribution. And I'm just going to take you over to the right and where we've done heat map modelling as a result of our measured data. And so the two levels that are shown here, 50 micromolar and 100 micromolar, and that map that you see, the black lines are blood vessels, and the rest of the space is the tissue. And you can see that at 50 micromolar in the plasma, that the tissue is not saturated. So even though the cells that are completely adjacent to the blood vessel wall will have healthy levels, as soon as you get further away from the blood vessel, those cells are not saturated. To get saturation across the tissue, we have to be in the order of 100 micromolar, which is plasma saturation levels. So that's what we aim at, and that's what part of my justification for our talks. So our recommendations to people are we recommend vitamin C intake around what we call the RDA. So all nutrients have an RDA or RDI, so recommended daily allowance or daily intake. And that number that we give out to people, it's important to know where that comes from. So all nutrients are thought to respond to this U-shaped curve with respect to benefit. So we all know too much of a good thing is bad for you. And essentially that's what this graph shows. So with the increasing nutrient intake, you come to a point where you lose the risk of deficiency. But then with more and more intake, you will reach a point where you have a rise in toxicity. Now this actual shape of the curve, or the distance between the RDA and the upper limit will vary for different nutrients, but essentially all nutrients will have a curve like that. Now where we set the RDA is at the point of where 90% of the population avoid deficiency. And we also have this other measure called the estimated average requirement, which is where half the population avoids deficiency. So when we make a recommendation for this about vitamin C, what matters here is how do we define deficiency? And so currently, deficiency is defined as scurvy. So rank deficiency, where you have a life-threatening imminent death. So the RDA for vitamin C is set at the point where 90% of the population will avoid rank deficiency. If that were another measure, and if we look here, so that's shown here. So with plasma levels for scurvy, that's a plasma level of 25 micromolar. So with 25 micromolar, you'll have 100% avoidance of scurvy. And that's what our aim is with the RDA. However, if we think back on those cohort studies and the epidemiological evidence that we have, then we're seeing increasing health benefits from vitamin C across the plasma vitamin C concentration up to plasma saturation. And so if we set 90% of the people avoiding or having optimal health benefit from vitamin C, then we would set the RDI at a level that gives us saturation levels in the plasma. Now, currently, that's not the case, particularly in New Zealand, in the US, and I think in Europe, some of the RDA levels have increased, but in New Zealand, we are currently 45 milligrams per day. This is dose of vitamin C per day with plasma concentrations, and so to reach saturation levels, you need at least 200 milligrams a day, and that's nowhere near anyone's recommended daily allowance. It is at the point where we see the association with global health improvement. So that's the number. You also see that our current RDA puts us on the very steep part of the curve. There's not a lot of room for manoeuvre either side of that. And in fact, if you fall anywhere slightly to the left of that, you're very much at risk of scurvy symptoms. So what's our population look like? Well, there's some data from the US, and we have some data from New Zealand as well. So this is a cohort of 400 normal, healthy New Zealand adults. So if we took, people in this room, we'd probably see a very similar distribution here, or maybe not, because maybe we all eat kiwi fruit. But this is very typical, where the average concentration for a normal healthy cohort's about 50 micromolar. Very few people are at the plasma saturation level. So only 20, maybe 30% of people are at the plasma saturation level. A considerable proportion are below that 50 micromolar, with actually quite a lot at 30 and below. And we know that those are the levels at which we're seeing symptoms of hypovitaminosis C. So there's a lot of room for improvement in terms of our vitamin C status. And the same thing has been found in the US as well. And so this is just looking at people who are, or sort of intake of micronutrients generally across the US population. Somebody was talking yesterday about measuring micronutrient levels and said the only thing we measure generally are the B vitamins. Interestingly, the B vitamins are the ones where we tend to get much better intake because they tend to come from meat. And so the ones that come from fruit and vegetables, the number of people who are below the average intake, which is half the deficiency level, it's really high. So the black bars are just the straight population, and the other bars show the effect of supplementation. So as you can see, that folate supplementation has hugely benefited people's folate intake. But essentially, we have, nearly half the population below even that half recommended level. So intake level, there's a lot of room for improvement here. So that brings me to why is this important? So why do we need to know? What do we know about vitamin C? And why do we think we should be aiming? What's underpinning that need for the plasma saturation levels? So we know that the basic facts, vitamin C is made in most plants and animals. So most animals don't have the challenge that we have. Their liver makes all the vitamin C they need, and they maintain their plasma at saturation levels no matter what. So, but we don't, because we are dependent on our intake. So, because we've lost this enzyme. We know that vitamin C, or ascorbic acid, is a very good reducing agent. So we talk about it as an antioxidant. We know it complexes with metals, and we know that it acts as a cofactor for a number of enzymes, and it's that metal binding capacity that allows it to do that. Now, just don't worry about the detail here, but we know that vitamin C is very labile. So when you have it in solution, it's very water soluble, but it reacts very quickly. It's destroyed very quickly by reacting with oxygen and by heat. So when you cook food, you start to destroy vitamin C, and when you leave it exposed to air, you will also lose activity. And it's that function, it's that property that we think makes it a very good antioxidant scavenger. So it's very readily oxidized. Whether that's an important in vivo function is another question. So we know all of those things in vitro. So if you take any solution of vitamin C, you put it into any oxidized, used as an antioxidant, a preservative for that reason, because it'll be the sacrificial molecule that reacts with oxygen before you get any oxidation of anything else. We know that in vitro, but we have very little information that actually happens in our bodies. And so, but having all of that information in vitro has led to the proposed antioxidant activity in vivo. Now, the problem with this is that it's very easy to draw these diagrams when you're writing papers or when you're studying the mechanisms of disease. It's almost impossible to measure those things in any living tissue. And so even in cells and culture, these things are really hard to measure because they're rapid reactions. And so it's the in vitro chemistry that's been extrapolated to a biological situation which we don't actually know that happens. So that's just an example. The best evidence we have for vitamin C being a target for oxidative stress is if we look in the plasma levels of people who are sick. So this is showing the differences between a normal population, all shown in white, their plasma concentration, and a cohort of people on admission to hospital. So these people all have various things wrong with them, but you see on admission to hospital, hospital patients have mostly below 25 micromolar plasma levels of vitamin C. So this is an indication that under stress, your body uses a lot more vitamin C and you actually need to increase your intake. Animals will increase their synthesis to compensate under these circumstances. Here's another example. These are sepsis patients. So these are people who are in critical care. And here's the normal population here. So with an average level of between 50 and 60. And you can see that mild or severe sepsis patients, they arrive in hospital with low levels, and with increasing time, their bodies are becoming depleted. And the more severely ill they are, the more depleted they become. So these are the best indications, we think, that vitamin C is a target for oxidative stress. Same across a range of diseases. So the black bars and the grey bars show levels below 25 micromolar. So people in hospital or with a range of diseases have high consumption rates of vitamin C. So what can we conclude from what in vivo evidence we have? We know that vitamin C turnover is accelerated during illness. We know that that's associated with disease severity. And we know that supplementation of those people, so those people who are ill, is associated with improved outcome. However, that doesn't tell us that that's an antioxidant activity. That tells us it's a target for oxidative stress. It doesn't say the reason that vitamin C is there in the body is to scavenge those oxidants. We have a whole orchestra of antioxidant activity that protects us from oxidative stress. And most of those things recycle. And so the one thing about ascorbate is once that oxidation happens, it has trouble recycling. So now I'm going to spend the remaining time talking about the other activities of vitamin C. So remember I said it gets taken up into all our different organs at different rates. So all our organs have different concentrations, and we think that actually correlates really well with the presence of these enzymes that depend on vitamin C for activity. And I'm going to talk about this family of enzymes because it's becoming hugely important, and there are some very exciting developments in this area. So they've got this terrible name, I'm sorry, the iron and 2-oxyglutarate dependent dioxygenases. Now they are everywhere in biology. All plants and animals have them. We need to think up another name, maybe. But they're commonly known in plants. They're the enzymes that are responsible for the plant cell wall. This is the reason that plants have vitamin C. This is why we get it from our fruit and vegetables, is that plants don't make it for our benefit, they make it for their own benefit. And so we're just passengers on that ride. So they need it for plant cell wall synthesis. We know that in our bodies we need it for collagen synthesis. Less commonly known that it's everywhere. It's in bacteria, it's in fungi, so involved in morphine synthesis and penicillin. There are hundreds of these enzymes. They all belong to the same family, and they all carry out the same difficult chemistry. And so that they have a reaction cycle that adds a hydroxyl group, so they need oxygen for activity, and they split that oxygen in their cycle, and they use that to hydroxylate. So they make a number of products. So this is a factory mechanism that makes a number of products in our body for a whole range of activities. So they all carry out the same chemistry. So they need a number of things to do that. They need oxygen from the blood supply. They need 2-oxyglutarate, which is an energy metabolite from the mitochondria in our cells. So this is a metabolic intermediate. They need iron. So this is one of the reasons, apart from your oxygen transport, that you need good iron uptake. And they need vitamin C to keep functioning. If that vitamin C is missing from the center of that diagram, these enzymes don't cycle, and so their activity stops. And so without the vitamin C present, they don't function. Why is that important? Well, we talk a lot about the activity of the collagen hydroxylase, and we all know that was the first enzyme discovered in this family. It was discovered in the 1960s, and it's responsible for stabilizing collagen, which is a major part of what holds us together. So apart from your bones, your collagen's the reason you're able to stand up. And that's what holds our tissues together. So it's a major structural protein. We know we need vitamin C for that. What we don't talk about as often are the other activities, and some of them are quite recent. Their hormone synthesis role has been known about 20 years, I think. There are a number of enzymes involved in that. More recently, in the last decade, has been the discovery of the HIF hydroxylases, which are involved in metabolic control. I'm going to talk a bit about those. In the last 5 years, we've discovered that epigenetic regulation, these enzymes are involved in the stabilization of the epigenome. We'll talk a bit about that. And there's also indications that they're involved in stress signaling. So all of these activities, what do they mean? What do they mean for us on a day-to-day basis? Well, what it means is that vitamin C is important in all of these things. We know this: tissue formation and wound healing, mood and energy, cancer prevention and treatment, immune system function, appetite control, gene function, or gene regulation, which controls pretty much everything, and our stress responses. So here's a global health story that is associated with one nutrient that we need on a daily basis. Now, as I indicated, there are other nutrients involved in these enzymes as well. So the support of this enzyme family underpins good health and longevity, as we know from the association studies. So I'm just going to give you, in the remaining time, some examples of the mood and energy, metabolic function and the gene function in association with vitamin C levels. Now the first example is the mood and energy example. This is just one of the molecules that is generated by vitamin C that's associated with mood and energy. And so this is carnitine, and this is the synthetic pathway, and where I've got the yellow stars, you'll see those two enzymes both belong to this family. They have iron, they use 2-oxyglutarate and oxygen. So they both belong to the same family. At 2 points in this synthetic pathway in our bodies, we need vitamin C to function. What does carnitine do? It's a molecule inside our cells in the mitochondria, so the energy factory of our cells, and it transports fatty acids from the cell cytoplasm into the mitochondria where they're available for energy. So if you don't have enough carnitine, you can't transport the fatty acids in for oxidative energy production. So absolutely essential to energy metabolism. Here's a nice little paper that was published in 2006, which demonstrates this really well with respect to plasma levels in a normal human population. So this is a study from Arizona, where they've taken a cohort of slightly obese people, or young people, they were, with a BMI around 30, I think, and they've stratified them according to vitamin C status. So what they've found is that most of them have very low levels, so these are their plasma levels, and so these are people obviously don't eat well, and their plasma levels 20 micromolar and below. And so they've taken these people and put them into two groups. One group they've given vitamin C supplement to, and after 4 weeks of supplementation, they've analysed their ability to metabolize fat and to produce energy. And so they've had these people walking on a treadmill, and then they've worked out from blood and plasma levels how much energy they've burned from fat and how much from protein. And so you can see here at week 8, so just look at the black bars, which are the average, the others are the individuals, that after week 8, half their cohort, so the placebo group, no increase. Here their plasma levels went up to 40 micromolar with their supplementation, but here's the interesting downstream effect. Prior to supplementation, these people burned little fat for energy when they were exercising. After the placebo group showed no increase, after supplementation with vitamin C, they start burning fat for energy. On the other graph, you see the reciprocal where they've been burning protein. So prior to supplementation, they're using protein for energy rather than fat. Now this was associated with the carnitine levels in their muscle. So they took muscle biopsies. They also found that these people, so the ones that were not supplemented, were tired after exercising. So they're unable to access the fat to burn for energy because their carnitine levels were too low. The next example that I want to give is the metabolic sensing. And so this looks terrible, I know, but there's in all our cells, there's a response to the lack of oxygen. So we know that if you go, mountaineering and you get altitude sickness, which is a result of decreased oxygen, there's all kinds of physiological responses to that. So our bodies are primed to survive low levels of oxygen for short amounts of time. And that's through the rapid response of this factor called hypoxia-inducible factor. It's present in all our cells, ready to go should we not be getting enough oxygen. And it's regulated by these enzymes that need vitamin C to work. What does this cofactor, what does this transcription factor do? It upregulates, in response to that, hundreds of genes that allow you to survive low oxygen. That's a good thing, mostly, but we need it to come on at the right times. And for that, we need optimal vitamin C in all our cells all the time. What happens when we don't have that? Well, hypoxia is a driver in a number of illnesses and disease states, and we know that hypoxia is a major driver in cancer growth. So this factor is one of the things that allows cancers to grow bigger than 2 millimeters, to outgrow their oxygen supply, and we know that vitamin C is involved in the downregulation of that. I'm not going to talk about that today. What I want to talk about is the effect on inflammation. My thing seems to have fallen off the side. And yesterday we had a lot about gut health. So we see on the left a normal intestine. The wall of the intestine is slightly hypoxic normally. So as it gets further away from the blood supply, you have moderate to low hypoxia, that's shown in red. If you have an inflammation, like you might have with any irritable bowel, then that hypoxia becomes extreme. And so you can see the huge red area over on your right. Does it matter? We also know that in that inflammation, there are lots of white blood cells. That's why they're there. They're there because there's an inflammation. Does that matter, and how does hypoxia manifest in those cells? So the cells we've looked at, and Anita's going to talk about these as well, are the white blood cells, or the neutrophil. So the neutrophil is the cell shown over here on the right. It's our primary defense against microorganisms. We have billions of these cells hovering around our body, wandering around our body every day. And they accumulate at those sites of infection, and they're exposed there to hypoxia. Now, and that's important, as it turns out. They need oxygen to function, but they need oxygen particularly for one specific function. And that is not necessarily to kill the bacteria, but to clear that infection. And so this is a lung infection. So when you have a cold or a flu, you know your lungs get congested. What they get congested with are these white blood cells. And and what we need for those to be removed. And we need for them to be removed safely, because they're full of really damaging molecules and enzymes that will cause havoc should they escape. What we also know, so that happens by a process called apoptosis, and macrophages, which are another inflammatory cell, they come and they ingest and remove these cells safely. But to do that, they have to recognize the signal on the surface of that dying cell. And what we know is that if these neutrophils don't have vitamin C, they don't undergo this process, and they don't show these signals. And so you see that happening over on the right, where the brown spots are the neutrophils, and the cells underneath are macrophages. And so you see, under normal circumstances, the brown spots will associate with the macrophages, and in fact they engulf them. If the cells are vitamin C deficient, they can't see those cells, and so they don't take them up. And that is the same phenotype as what you see in hypoxia. And so these are the associations we make between vitamin C and that HIF-1 activity. I'm just going to very quickly talk about epigenetics, because this is the major focus for the future, I think, with respect to diet. So if you will have heard of epigenetics, and essentially it's the overwriting of our genetic code. So we know we have genes, but we need to determine when those genes are being read and when they're not. So there are enzymes that add marks to our genes that add the signals for switching off, and there are enzymes that remove them, and so they're called erasers. So these eraser enzymes belong to this family, and they are the iron and 2-oxyglutarate-dependent dioxygenases, once again, and there are dozens of them that are involved in epigenetic regulation. This is really new information. We don't fully understand epigenetics, but we know the implications of it. This is where nature and nurture combine, and we have vitamin C being involved in the very fundamental processing thereof. So you see that on the right. So if you just look at in a cell line that's been given vitamin C or not, the dots show the different gene expression. And so the gene expression in those cells is completely different when they contain vitamin C or not. So if you don't have enough vitamin C in your cells, you've got a completely different cell and a completely different genotype. This is important for cancer, it's important for development, it's important for inheritance. It's going to be a really big area. So this is our take-home message then with respect to these enzymes. We need things from our diet to support the enzyme activities. And in fact, we get this from a good healthy diet. So we get iron from our meat and proteins. We get metabolic intermediates from fruits and grains. We get oxygen from fruit and vegetables by improving blood supply. And we get vitamin C as well. And we need all of those things to support these enzyme activities. So these are my take-home messages. So just to remember, vitamin C levels are positively correlated with health and survival, and our body stores and tissue saturation are dependent on an adequate plasma supply. So vitamin C is depleted by oxidative stress conditions at intracellular levels. It's the intracellular levels that influence the activity of these enzymes, and many of the physiological benefits are going to reflect the cofactor activity of vitamin C rather than the antioxidant level, we believe. So I just want to thank these people, and I want to leave Anitra enough time to talk. Thank you.